Assalamu alaikum. In this lecture, we'll go over biochemical genetics, diseases related to metabolism, and genes associated with these disorders, and modes of inheritance. Metabolic disorders that humans are born with are known as inborn errors of metabolism. So it's a collection of metabolic diseases. Now these are basically inherited enzyme deficiency disorders leading to a disruption of normal bodily metabolism, leading to a pathological conditions. Now some of these can appear at birth, others can appear later on in life. Now what happens is that during metabolism you have conversion of one substrate to a product which can be a substrate for another enzyme, resulting in the production of the final product. Now what happens is that during this pathway, if one of the enzymes is defective and it's not functioning well, then what happens is that you will have accumulation of the substrate. You will have a reduction in the in the product, and there might be sort of like a pathway by which the substrate, one of the substrates, one of the intermediates in this pathway can be converted to a toxic metabolite. So all of these can result in cell toxicity, cell death, and resulting in whatever deficiency depending on the affected organ. So there are different types of inborn errors of metabolism. You have silent disorders, which are not life-threatening disorders, but if left untreated, they can result in brain damage as well as developmental disabilities. An example is phenylketonuria, and we'll talk about this as our first example later on. Now we have other types of inborn errors of metabolism that can cause acute metabolic crises, and they are life-threatening. Examples include disorders related to the urea cycle. Now what happens is that as long as they are not born in the fetal stage, they are protected in utero. Okay, yeah, as a result of the maternal circulation providing the infant or the fetus with the necessary products. But as soon as they're born, they would immediately, within few days, months, they would be affected by the reduction in the presence in the they would be affected by the absence of the important product as well as accumulation of the substrate or production of the toxic metabolite. Now we also have inborn errors of metabolism that can cause neurological deterioration. Examples include the Tay-Sachs disease as well as Gaucher disease, and we'll talk about these at the end. So basically, in terms of the genetics of inborn errors of metabolism, they usually follow the one gene, one enzyme concept or hypothesis, indicating that each gene is produced by a single gene. So that was like contrary to the to bacterial genomics, where you do have operons or one gene that can produce multiple enzymes. Anyhow, so usually mutations result from single base pair substitution as well as deletion and insertion. Now they can be heterogeneous. A person may suffer from incomplete penetrance. Okay, so basically the symptoms are variable among individuals, and there are racial and ethnic variations as well. Okay, so at least in our region right here, consanguinity plays a major role in the appearance or the manifestation of these disorders, and it increases the risk of having such condition by 3 to 5%. Now, Usually the mode of inheritance is autosomal recessive. Males are, um, if the male to female ratio can determine if it is an autosomal disease or sex-linked disease, specifically X-linked condition. And usually it is a recessive type of inheritance rather than dominant. There are some exceptions though. Now, there are some X-linked recessive disorders, such as the Lesch-Nyhan Syndrome, and there are very few are autosomal dominant trait. Now, how about treatment? Treatment is basically the first thing is dietary restriction. So the substrate, the intake of substrate is limited so that there would not be accumulation of the substrate or production of the toxic byproduct. Patients can be supplemented with the deficient product. There might be stimulation of alternative pathways to produce that product. There could be patients could be supplied with vitamins and cofactors. Organ transplantation is an option, but it doesn't work for all conditions, and its success rate is quite variable. Now, in terms of enzyme replacement therapy, it hasn't been proven efficient for many clinical conditions, and again, its success is also variable. Now, gene therapy is an option, but it hasn't been proven successful for many of the conditions. Now, the inborn errors of metabolism can be categorized into different types. You can have disorders of protein metabolism, such as amino acid metabolism. You can have disorders in carbohydrate metabolism, and there are different types. There are disorders related known as lysosomal storage disorders. You can have fatty acid oxidation defects, lipid metabolism in general, mitochondrial disorders, as well as peroxisomal disorders. So there is a large array of disorders that can be linked to inborn errors of metabolism. Now, we're not going to talk about all of them. I'll just try to talk about some of the major ones that can be relevant to us and to you as you grow up in your career. Now, so these disorders in general have different manifestations depending on the pathway that is affected. So disorders of carbohydrate or protein metabolism, as well as disorders related to energy production, they tend to be present in, or to present in the neonatal period, as well as early infancy, and they tend to be rapidly progressive. Okay, so less severe variants may present later in infancy or childhood and tend to be episodic. Remember that they follow the incomplete penetrance pattern of inheritance. Now, on the other hand, fatty acid oxidation and lysosomal storage disorders are more subtle. Okay, so they're more subtle in early in life, but then they become progressive and quite manifested during adulthood or until the patient reaches adulthood. Now, autosomal inborn errors of metabolism, the defective protein or enzyme is usually diffusible, and there could be some loss of function of the enzyme. So sometimes it's not really total loss of enzyme activity. Okay. Now, on the other hand, there are some enzymes, especially those that are rate limiting, meaning that these are the highly regulated enzymes, or enzymes that are formed from multiple complexes or multiple enzymes, like pyruvate dehydrogenase complex, for example. Loss of one of the alleles or reduction in the production of enzyme can be manifested in the heterozygous state, and they usually follow a dominant inheritance as a result of that. So let's talk about some of the disorders related to inborn errors of metabolism. We'll start with amino acid metabolism. Now, phenylketonuria is one of the early conditions that have been known in terms of etiology. That is the cause of the disease. So it's basically you have a problem in the conversion of phenylalanine to tyrosine, okay, as a result of defective phenylalanine hydroxylase. Now, the thing is, if there is no tyrosine, then there would be accumulation of phenylalanine, and phenylalanine can be converted to another product known as phenylpyruvate or pyruvic acid. Now, accumulation of phenylpyruvate may be toxic to the central nervous system, okay, as well as the accumulation of phenylalanine, and that can cause intellectual impairment. Now, children usually have blonde hair and blue eyes, and the reason is that there would be deficiency in the amount of tyrosine, which is important for the production of pigments, and we'll talk about this later on in albinism. Now, there's usually now there is screening for phenylketonuria in born children. So as soon as they are born, blood spots can be taken from their heels, and any deficiency of phenylketonuria can be diagnosed early on. And once diagnosed, these children can be treated by lowering the intake of phenylalanine. Another disease that I would like to talk about is Alkaptonuria. Now, the thing with this disease is also related to the metabolism of phenylalanine and tyrosine, except that the defective enzyme is known as homogentisic acid oxidase, which converts homogentisic acid to the final product, which is mallyl acetoacetic acid. Now, if this enzyme becomes defective, there would be accumulation of homogentisic acid, which accumulates in urine. It is excreted in urine. It accumulates in the body, and it can precipitate in certain places or certain tissues, such as it can be found in earwax, cartilage, joints, and this can lead actually to painful arthritis later on in life. Now, this can be detected. Deficiency of this enzyme can be detected by exposing the urine to air, and that results in its oxidation and conversion to a dark color, brown black color, and you can see the accumulation of the homogentisic acid in tissues. Now, I like to talk about this disease because it is common in one of the villages in Karak, and one of my colleagues started investigating this disease further, and he identified a number of novel mutations in the gene of this enzyme. Another condition that I'd like to talk about is albinism. Now, albinism is related to deficiency in the enzyme tyrosinase. So what happens is that there would be failure to convert tyrosine into the final products, eumelanin and phenylmelanin. Now, these two products are pigments really, and they give our skin and hair the colors. So eumelanin is present in high quantity among black individuals, and of course, it goes down as you go up in latitude. Now, phenylmelanin gives people the red pigments, red hair, skin, and so on. Now, there are different types depending on the severity of the disease. Type 1 ranges from no to residual enzyme activity. There is this metabolic condition that we talked about before, and we said that it is found in the Amish people who live in Pennsylvania. Now, this condition right here results from deficiency in the enzyme branched chain keto acid decarboxylase, which is necessary for the metabolism or catabolism of branched amino acids, valine, leucine, and isoleucine, and these amino acids are essential amino acids. So these amino acids tend to accumulate in blood and urine as well, and they give urine an odor that is similar to that of maple syrup, and that's why they call it maple syrup urine disease. Maple syrup is a syrup that we use when we eat for breakfast waffles or pancakes. Now, all of the previous diseases are autosomal recessive diseases, so you need to pay attention to that. Now, metabolic disorders of nucleotide metabolism, again, there's a wide range of disorders that have been identified. It's really hard to talk about all of them, so I'm going to pick a few of these disorders. Now, we'll talk about the Lesch-Nyhan Syndrome. The Lesch-Nyhan Syndrome is related to defective enzyme hypoxanthine guanine phosphoribosyl transferase. Now, this enzyme is responsible for converting hypoxanthine and guanine, so catabolism, or sorry, the conversion of hypoxanthine into inosine monophosphate and guanine into guanosine monophosphate. Now, if this enzyme is defective, you would have accumulation of guanine and hypoxanthine, which would be converted eventually to uric acid. And uric acid can accumulate in joints, and it can be related to nupros. Nupros is gout. Now, it can be treated partially with allopurinol, which is an inhibitor of the enzyme xanthine oxidase, resulting in reduction in the accumulation of uric acid in the body. But it has variable success, and it doesn't work 100%. Now, let's talk about disorders of carbohydrate metabolism. What I'm trying to do here is I picked the disorders that are either important or ones that are not covered in other courses. So there are disorders related to metabolism of fructose. The first one is essential fructosuria, and the other is hereditary fructose intolerance. So with the essential fructosuria, there is a defect in the enzyme fructokinase that converts fructose to fructose 1-phosphate. Now, this is a benign condition. However, there will be accumulation of fructose in the urine. It is an autosomal recessive disorder. Another condition is the hereditary fructose intolerance, which is also an autosomal recessive disorder. It's related to deficiency in the enzyme aldolase B, resulting in trapping of fructose 1-phosphate in cells. Now, this can cause hypoglycemia, vomiting, jaundice, a number of symptoms. So these individuals should reduce their intake of fructose and fructose containing products like chocolate and gum and anything related to that, so as well as sucrose. Now, intake of these can result in hepatic failure as well as death. So now, another condition that we should focus on is classic galactosemia, which is related to deficiency in the enzyme galactose 1-phosphate uridyl transferase. Now, it is an autosomal recessive disorder, um, and it can cause galactosemia and galactosuria, vomiting, diarrhea, and jaundice as well. Now, what happens is that there will be accumulation of galactose 1 phosphate in several tissues, affecting them, of course. Now, prenatal diagnosis is possible, um, and the therapy basically, again, is reduce intake of galactose in the body, as well as, of course, lactose in milk. Despite that, developmental delays can result. Okay. So, in terms of lipid metabolism, there are many disorders, but what I'll talk about in here is sphingolipidoses. Now, sphingolipidoses basically are related to problems in the metabolism of sphingolipids. Now, sphingolipids are important in many tissues, particularly in the central nervous system. So, genetic disorders usually they are manifested by neurological pathologies. Now, many of these disorders are related to the degradation of sphingolipids, which go through stepwise enzymatic reactions. Now, in general, they are autosomal recessive, except for one of them known as Fabry's disease, which is an X-linked disorder. Now, there's a list of sphingolipidoses. Do not memorize this table. You can just look at it and read it. But we'll focus on a number of disorders. So the first one is Tay-Sachs disease, which I mentioned before in one of the previous lectures, and I said that it is common among Ashkenazi Jews, and we mentioned why. Now, this is caused by defective enzyme known as beta-hexoaminidase, which would result in the accumulation of GM2 gangliosides. So you can see the stepwise degradation of sphingolipids from GM1 gangliosides to GM2, GM3, and so on. So problems in the enzyme beta-hexoaminidase would result in accumulation of the GM2 gangliosides. Another condition is known as Niemann-Pick disease, which is related to defective sphingomyelinase. Now, this enzyme converts phosphatidylcholine in sphingomyelin into ceramide. So, deficiency of this enzyme would result in the accumulation of sphingomyelin in liver and spleen. And there are different types, again, according to severity of the condition. Now, another common sphingolipidosis, one of the common sphingolipidoses is known as Gaucher's disease, which is related to deficiency of the enzyme glycosylceramidase or glucocerebrosidase. Now, deficiency of glucocerebrosidase would result in the accumulation of glucocerebroside in lysosomes. And again, there are different types according to severity as well as frequency. Let's talk briefly and quickly about multifactorial diseases. Now, since for multifactorial conditions, particularly polygenic conditions, several genes are involved in the etiology of these conditions. It's difficult to pinpoint the ones that are associated or responsible for these diseases. So as a result, scientists have initiated a number of initiatives, projects such as the HAP-MAP project. It's also known as the 1000 Genomes Project, which is basically sequencing the DNA of 1000 individuals from different ethnic groups and looking at associations between certain markers, particularly single nucleotide polymorphisms with conditions and phenotypes. Now, many of these single nucleotide polymorphisms are in linkage disequilibrium. Now, what this means is that they are co-inherited with certain other markers or genes. And this is what linkage disequilibrium means. It means it's basically non-random association of alleles at two or more loci in a general population. So they would say that this particular SNP is associated with this gene or this phenotype in a population, and this SNP is associated with that disease or that condition or that trait or that phenotype. So when alleles are in linkage disequilibrium, so it defies the Hardy-Weinberg equilibrium that we talked about in the previous lecture. When alleles are in linkage disequilibrium, we say haplotypes do not occur at the expected frequencies, indicating, so it's not random. Their association is not random, indicating that their association to a certain genotype or phenotype, and so on. So these regions of linked SNPs are known as haplotypes. And the International HapMap project was set to identify SNP frequencies and haplotypes in different populations, and the database is freely available. Technology has improved quite a bit in the past 15 years, to the extent that we can easily analyze or sequence the whole genome of individuals and populations. So this has generated a new field or a new type of studies known as genome-wide association studies, or GWAS, where basically you would have analysis of DNA by sequencing and trying to find association between markers like single nucleotide polymorphism to phenotypes and other genotypes. So there are about 10 million SNPs in our genome. About 500,000 to 1 million are relevant. These are the ones that are found in or present in relevant parts of the genome, or that can be linked to certain genotypes and phenotypes. Using a DNA microarray known as SNP chip, about these SNPs can be studied all at once. Now the thing with GWAS or these studies is that it is hypothesis free, meaning that they would fish for certain markers, genetic markers that are associated with the genotype, without any prior knowledge of or bias into the association of certain SNPs to these genotypes. Okay. So basically relevant and important information can be generated, except that with these studies large number of subjects must be involved or must participate in such studies. That's one, and that requires a new type of statistical analysis. So basically you studied in statistics that anything with a p-value of less than 0.05 would be significant, right? Except with GWAS, this is not really beneficial, because you would get like thousands of SNPs associated with certain phenotypes. Rather, the p-value has become 5 times 10 to the minus 8. Okay, not the typical 0.05. So now, the thing is, for most traits, studies have shown that each genetic marker is responsible for this trait by a small fraction. So for example, you would find in terms of height, let's say, you would find that a certain genetic marker is associated with an increase in a person's height by 1 millimeter. You would have another genotype that would contribute about 2 millimeters to a person's height, and so on. So there is little contribution for all of these markers, and collectively they play a significant role. Now, yes, we have identified a number of genetic variants, and each one of them can contribute a small portion of the trait, but there are also rarer variants that are that have not been identified yet, and they can explain some of the missing heritability, that is, the missing genetic contribution to a certain trait. I'll give you an example from something that I've been working on, which is multiple sclerosis. Now, this is a heterogeneous neurological autoimmune disease. It is an inflammatory disease that results in the degradation of the myelin sheath. Now, it has been known that there are a number of factors associated with multiple sclerosis, and these include genetic factors, as well as environmental factors and immune regulation, or even pathogenic contribution as well. Okay, so each one of these factors, if you go back to the threshold model that we talked about in multifactorial inheritance, each one of these can contribute a little bit to the development of multiple sclerosis. Now, look at the contribution of environment. So this is the distribution of multiple sclerosis globally. You can notice that there is the frequency of MS is very high in higher latitudes, right? And it decreases as you go south, as you go towards the equator. And it has been assumed that this is due to exposure to sun and probably deficiency of vitamin D. And it has been found really that vitamin D plays an important role in the development of MS. So deficiency of vitamin D can lead to the development of multiple sclerosis. Now, the thing is, genetic studies have identified a human leukocyte antigen, a certain human leukocyte antigen to multiple sclerosis, and that was in 1973, long time ago. And it took scientists 30 years to identify additional genes. So in 2007, 2 genes were found to be associated with multiple sclerosis: the interleukin 7 receptor gene and the interleukin 2 receptor gene. And the reason behind this discovery after all of these years is not only the development of newer and better technologies, but rather the involvement or the participation of large number of MS patients in such studies. Previously, hundreds of MS patients were analyzed. The genomes of hundreds of patients were analyzed, but in these studies, about 1,000 or over, about 1,000 plus and minus parts were analyzed, and that's why these 2 genes were identified. So it was realized that that larger number of subjects should be analyzed or should participate in such studies. So after a few years, about 10,000 individuals participated in a study identifying 55 single nucleotide polymorphisms. Later on, more and more studies were conducted, and now we know that there are about 200 variants, 200 genetic variants, 200 markers, 200 single nucleotide polymorphisms to be associated with multiple sclerosis. Such studies, the GWAS, the studies generate something that looks like a Manhattan plot. Now, with the Manhattan plot, you have the chromosomes right here. chromosome 1, chromosome 2, chromosome 3, and so on. And here you have the location of the different SNPs. And then analysis would indicate which SNP is important in these to that disease. Okay, so you get something that looks like a Manhattan skyscraper, and that's why they call him Manhattan plots. So you notice that this one right here passes the threshold of the statistical value that we just talked about. So you can see that this gene is actually the HLA region related to this particular one, HLA-DRB1. You can see the involvement of a number of genetic factors or SNPs or markers, including the interleukin-2 receptor gene shown in here. So this is really the future of such studies, and hopefully we will find out the mechanisms, the causes, the genetic causes of the certain diseases that are related to more diseases such as multifactorial diseases such as diabetes and so on.